Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CSIR Net JRF Life Science question paper of Shift 2 Part C. Questions are discussed. The exam was conducted in the year July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR Net JRF being discussed, you can go to my channel Omni Bio Entrance. In the playlist, select CSIR Net JRF, you will find them there. First question. The feedback control of the branched amino acid biosynthesis pathway in Arabidopsis is given below. So pyruvate to OIV, then to leucine. Leucine and valine, they will inhibit AHAS. Leucine will inhibit IPMS only. Okay. The activity of acetohydroxy acid synthase enzyme is feedback inhibited by leucine and valine synergetically, whereas isopropyl malate synthase enzyme activity is inhibited by leucine only. I told you just now. Feedback resistant mutant lines. Now what are that? Feedback resistant mutant lines. Feedback resistant mutant lines, they are organisms that are mutated, so they are resistant to feedback inhibition. Feedback inhibition is not going to affect these organisms. Of AHAS and IPMS genes are AHAS21D and IPMS121D respectively. The phenotype of these feedback resistant mutants was analyzed by growing them in the following Murashki-Skoog media combinations. Okay, now which media combination these organisms are going to grow? So MS medium, they will grow. Both of them will grow in MS medium. Both AHS and IPMS resistant lines are going to grow on MS medium. Now what about AHAS? AHAS21D it will grow on MS medium only, as well as MS medium supplemented with valine plus leucine. See here, valine, leucine, they inhibit AHAS, but this is a mutant. That means feedback inhibition by valine and leucine is not going to affect the AHAS, so it will grow in medium supplemented with valine and leucine. So A will grow. Sorry, AHAS21D will grow in A and C. Now in which one B will grow? B will grow in MS medium as well as MS medium supplemented with leucine only, because IPMS11D is a mutant. So even when leucine is present, the enzyme will not get inhibited. So it will grow in leucine. So IPMS11D will grow in A and B. So that gives your answer as option 3. AHAS21D will grow in both A and C, and IPMS11D will grow in both A and B. Then next one. Results of immunoprecipitation of HA-YFG are shown below. Given below are options of the controls that could be used to confirm that F1B actually associates with HA-YFG. So these are the various controls that are mentioned in the options. From that you have to choose the most appropriate control or the controls. Okay, now what is immunoprecipitation? It is a technique that uses antibodies to isolate specific antigens from a mixture of antigens. Now the antibody will be added to this mixture containing the antigens and incubated, and you will get antigen-antibody complexes. So from the antigen-antibody complexes, you want to isolate a specific one. So you will have a solid phase matrix with agarose beads. The antibody antigen complexes will be passed through this column. Then it will be washed and eluted and analyzed using SDS page and Western blot. So the question here, it is asking for a control lane. So include a lane where alpha HA is not added but protein A saccharose is added. Okay. Alpha HA is not added but protein A saccharose is added can be used as a control. A can be used. Include a lane where neither alpha HA nor protein A saccharose are added. That is already there over here. Include a lane where alpha HA is added but protein A saccharose is not added. Now protein A saccharose is not added means you cannot isolate your target protein of interest, because you will pass the immunoprecipitate through the column where it will bind to specific ligands in the column. So your target protein also will bind to the ligands in the column and it will be eluted out. So that cannot be carried out if you protein A saccharose is not added but alpha HA is added. So C cannot be used. Include a lane with alpha MYC is added instead of alpha HA before addition of protein A saccharose. That is also can be used. So A and D can be used. So answer is option 3. Then next one. Eukaryotic transcription factors TF1 and TF2 bind to independent cis regulatory elements cis1 and cis2 upstream of Tata box and positively regulate gene expression. So this is that cis1, cis2, this is Tata box, this is your gene. So TF1 will bind to cis1, TF2 will bind to cis2. Now histone modifier HM1 binds to TF1 but not to TF2. That is over here. TF1 it will bind HM1 but it will not bind to TF2. In order to determine how genes are regulated by these three factors, an in vitro transcription translation assay was set up. A packaged DNA containing regions from cis1 to cis2 along with eukaryotic minimal promoter fused upstream of a luciferase gene was purified. So to do this analysis, what you did was you purified cis1, cis2, then promoter, then luciferase gene under the control of the promoter. Okay. And you check for the luciferase activity upon addition of TF1, TF2 or a combination of TF1 and TF2 and HM1 in the presence of RNA polymerase and translation mix. So what do you get? You got a graph there. This is the graph. Here you see RNA polymerase is added in all, translation mix is added in all, but TF1, TF2, HM1 you are either adding or not adding, and you check for the luciferase activity. So here if you see, upon addition TF1 is absent, luciferase activity is less, here also, here also, here also, but upon addition of TF1 luciferase activity increases here and here. Here also it increases. Here it is increased. All are there. Then TF2 if you don't add, but TF1 is present, luciferase activity increases. But if TF1 is absent and TF2 is present, luciferase activity increases. So showing that TF1 is independent of TF2. Both are absent, luciferase activity is less. TF1 is present but TF2 is absent, again luciferase activity is more. Okay. Then HM1 also in the same way. Now we will read the statements. TF2 activates luciferase expression independent of TF1 and HM1. No. Because wherever TF2 is present alone, without TF1, luciferase activity is less. If you see here, TF1 is absent, TF2 is present, luciferase activity is less. Okay, again TF1 is absent, TF2 is present, luciferase activity is less. So TF2 activates luciferase expression independent of TF1 and HM1 is wrong. Then HM1 activates binding of either of TF1 or TF2 to their cognate cis elements to activate luciferase gene expression. That is also not correct, because when HM1 was alone, when HM1 here, when HM1 was alone without TF1 and TF2, luciferase activity was less. With TF1 present and HM1 present, luciferase activity increased. With TF2 alone and HM1 alone but without TF1, again luciferase activity decreased. Okay. So HM1 activates binding of either TF1 or TF2 to their cognate cis elements to activate luciferase gene expression is not correct. TF1 binds to cis1, that is given in the question. Recruits HM1 to modify DNA allowing TF2 to bind CS2 to enhance luciferase expression. That is correct. Recruits TF1 binds to cis1. TF1, it will bind to cis1. Okay. Recruits HM1. So TF1, it will recruit HM1 and activates to modify DNA allowing TF2 and to bind cis2, that is given in question, to enhance luciferase expression. Yes. Here if you see, here both TF1 and TF2 are present but HM1 is not present. Luciferase activity is there but it is less when compared to when all three are present. Okay. So TF1 binds to cis1, recruits HM1 to modify DNA allowing TF2 to bind cis2 to enhance luciferase action. TF2 binding will happen only when TF1 is there. Otherwise TF2 will not bind. So TF1 will bind to cis1, recruits HM1. HM1 will also bind only when TF1 is bound to cis1. Okay. So 3 is the answer. Now what about 4? TF2 binds to cis2, recruits HM1 to modify DNA allowing TF1 to bind cis1 to activate. No, that is not correct. 4th is not correct. Then single-stranded DNA binding proteins of 3 DNA repair proteins. Okay, single-stranded DNA binding properties of 3 DNA repair proteins was investigated. So you have got a single-stranded DNA. The biotin-ylated single-stranded DNA. This is the biotin label for single-stranded DNA. Label for single-stranded DNA was prepared and incubated with proteins in different combinations as shown below. So this single-stranded DNA which was biotin-ylated, it was incubated with different proteins, either A or B or C, or a combination of A and B and C was also incubated. This was followed by streptavidin pull down. The streptavidin, it will bind to the biotin label and it will pull down the single-stranded DNA bound proteins which will be detected by Western blot analysis using specific antibodies. Which of the following statement is not a correct conclusion from the above study? Okay. Now when protein A, B, C are not present and single-stranded DNA binding DNA is added, you don't get presence of protein A, B, C in the Western blot. But when protein A is added to single-stranded DNA binding single-stranded DNA, which is biotinylated, you get a band of A showing that protein A binds to single-stranded DNA. But when protein B is added, you don't get a band. Protein C is added, you don't get a band, showing that B and C don't bind to single-stranded DNA. But when a combination of protein A and B is added, you get two bands, showing that B must be interacting with protein A. Because you didn't get a band when B alone was added to single-stranded DNA, you didn't get a band there, showing that B doesn't interact with DNA. But it interacts with DNA indirectly by binding with A. That is what is seen in this lay. But when C is added, what will C do? No bands are obtained. So C is inhibiting the binding of A to the single-stranded DNA. B doesn't bind to single-stranded DNA. So what is binding to single-stranded DNA? It is A only. So C is inhibiting the binding of single-stranded protein A with single-stranded DNA. Now see the statements. Protein A binds to single-stranded DNA. Yes, this lane. That is correct. Protein B does not bind to single-stranded DNA. That's also correct. You didn't get a band over here. Protein C destabilizes the binding of protein A to single-stranded DNA. Yes. 3 is also correct. Protein C interacts with protein A but not with protein B. If that was the case, when C was added, you should have got a band over here in this lane. But that is not present. So 4 is the incorrect statement. And they have asked for the incorrect statement. So 4 is your answer. Then next one. The following 4 DNA oligos are mixed in equimolar concentration, heated to 95 degrees and slowly annealed in a microcentrifuge tube. Which of the following secondary structures will predominantly be formed or observed? 2 separate B forms of double-stranded DNA structure. If you see these DNA sequences, you can see that there are no complementary sequences to form DNA-DNA interaction. Just like in B form of double-stranded DNA. So first one is incorrect. 4 individual stem loop DNA structures. Self-complementary sequences has to be present for the formation of stem loop DNA structures. That is not there in any of the DNA sequences. So 2 is also not correct. 2 separate Z forms of double-stranded DNA structure, that is also incorrect. 4-stranded holiday junction, just like you see in this picture, it will be obtained. So 4 is the right answer. Then the surface electrostatic potential map of an 18 kDa protein is shown below. Shades of the blue and red on the surface denote positively and negatively charged surface respectively. Which one of the options represent the most likely natural substrate for the protein? So this is positive and negative charges. So blue is positive, red is negative. Fatty acid and cellulose. Cellulose doesn't carry any charge. It is only a polymer. So that is not answer. DNA and RNA, yes, they will bind to the positively charged blue region because they are negatively charged. So DNA and RNA is the answer. DNA and cellulose, no. Only cellulose, no. So answer is 2. DNA and RNA. Very simple question for part C. Then next one. Isolated mitochondria were either treated with protease or briefly incubated in hypotonic solution prior to treatment with protease. The reaction was stopped and the samples were probed for the presence of MTG2. Porine at the outer membrane, cytochrome C in the intermembrane space, KDH in the matrix using Western blot analysis. Based on the gels above, MTG2 is localized in. So where is MTG2 localized? Now if you see intact mitochondria, you can see that porine, upon increase of the protease concentration, porine is getting degraded. Similarly, cytochrome C also getting degraded because band intensity is getting less. But MTG2 is remaining as such. KDH is also remaining as such. In mitochondria placed in hypotonic solution also, porine is getting degraded when protease concentration increase. So here also band intensity is less. Similarly, cytochrome C is also getting degraded. But here also MTG2 and KDH are remaining the same. They are not increasing. So MTG2 located in a place that is not accessible to the protease. So KDH is also located in the same place. So where is KDH located in the question? In the matrix. So MTG2 is localized in the matrix. Answer is option 3. If it was in the outer membrane facing the cytosol, that is first option. Porine is located at the outer membrane, it got degraded on increasing concentration of the protease. If it was in the intermembrane space, cytochrome C, which is in the intermembrane space, got degraded in both hypotonic as well as intact mitochondria. So again it is not the correct. Traversing the inner and outer membrane, that is also not correct. So it is in the matrix. Now next one. A uniformly labeled P32 single-stranded DNA was incubated with a homologous double-stranded DNA. So this is single-stranded DNA. It was incubated with homologous double-stranded DNA in the presence of RAD51 or an RPA along with ATP or non-hydrolyzable ATP gamma S. So RAD15 was supplied, RPA was supplied, ATP was supplied, ATP gamma S, which is non-hydrolyzable, was also supplied. To study the 3-strand exchange reaction, the reaction were terminated at various time points. DNA were digested with echo R1 followed by electrophoresis and autoradiography. So this is the echo R1 site in the 5' 3' direction. Results are as shown in the figure. Okay. Now what is this 3-strand exchange reaction? Now that is seen in, it is a step in the homologous recombination in repair mechanism. Okay. Now what will happen here is a recombinase will bind to the single-stranded DNA to form right-handed helical filament. This one. Then this helical filament, it will bind to the double-stranded DNA to form a synaptic complex. Then the recombinase, it will facilitate strand exchange between the single strand and the double-stranded DNA, creating a new heteroduplex. That is this. This is the new heteroduplex. And the reaction, it is catalyzed by RAD51 and ATP in eukaryotes. Now see the statements. Based on the above data, which of the following statement is incorrect? RAD51 require ATP hydrolysis for strand exchange reaction. Now RAD51 ATP, you get all the 3 bands, 1 kb, 4 kb, 2 kb at 60 minutes. But when ATP is not there, but ATP gamma S, which is non-hydrolyzable, is present, you don't get any bands, showing that RAD51 requires ATP for its reaction. Now if again at 60 minutes you supply RAD51 and then ATP without RPA, you don't get any bands, showing that all 3, that is RAD51, RPA and ATP are required for the functioning of strand exchange reaction. Okay. So first one is correct. So that is not the statement what we want, because they have asked for the incorrect statement. Then second one, strand exchange does not take place in the absence of RPA. Yes, if RPA is absent but ATP and RAD51 are present, strand exchange doesn't take place. You don't get any bands there. The polarity of the strand exchange reaction is in the 3' to 5' direction. That is incorrect, because if you see the 10-minute reaction, you see that 1 kb has exchanged. Then in 30 minutes it is 4 kb and 1 kb. Then 60 minutes all 3. And again at 90 minutes all 3. Only the band intensity has changed of the one which is 2 kb, because 2 kb has completely transferred by it is have exchanged by the time it is 90 minutes. So it is in the 5' to 3' direction. So this order, that is 1 kb first, then to 4 kb, then 2 kb, that order is the same as this in the 5', 3' direction, that is 1 kb, 4 kb, 2 kb. So the polarity of strand exchange reaction is in the 5' to 3' direction, not 3' to 5' direction. So they have asked for the incorrect statement, so 3 is the answer. The rate of strand exchange is 7 kb per hour. Yes, that is correct. Strand exchange, strand is 1 plus 4, 5 plus 2, 7. 7 kb strand. Per hour, it is at 60 minutes. Here also 60 minutes. At 60 minutes, all 3 strands are exchanged. See here, you get all the 3 bands. So it is 7 kb per hour. So that is also correct. Then next one. The following statements are made about regarding formulation of the advanced candidate anti-malaria vaccine RTSS. So RTSS, it is a malaria vaccine also known as Mosquirix, and it contains circumsporozoite protein, that is CSP, a truncated form of the major surface antigen of the Plasmodium falciparum sporozoite. What is that? It is a circumsporozoite protein, that is a truncated form of the major surface antigen of the Plasmodium falciparum sporozoite, as well as hepatitis B surface antigen. So here only option C is correct, all the others are not correct. Answer is option 4. Then next one. A researcher wants to switch 2 fragments A and B shown in the figure below, A, B, using PCR. She uses primers pairs A and B, A, B, to amplify DNA A, this DNA. Thereafter, she mixes equal concentration of PCR amplified A and DNA B to set up a PCR using primers A and C. Okay, the plus strand sequence of the two DNA fragments are given below. So this is the plus strand sequence of A. This is that of B. And which of the following option represent the correct sequence of primer B? So we have to take A only into consideration, because in A only you have got the primer B. So this is the A sequence. I have rewritten it here. This is the primer sequence for B. 3' to 5'. Okay. This way. So it will be ending with TC, 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 5' to 3' or CT, 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 3' to 5'. So that is seen only in option 2 and option 3. Now which is the sequence over this side? This sequence you have to see which one it is. Here it is AG, TC, GA, CC, TG, this region mostly. Okay. Must be this sequence. But here if you see in option 3, it is AGCTT GCA, that is same as the one in B. We are not considering B here because that is not asked in the question. But why they have given the B sequence here, I don't know. But anyway, 2 is the answer. Then next one. A DNA sequence is given below. This is the sequence. The following primers were designed to amplify the above sequence. If we negate the effect, negate means we don't consider the effects of primer length, TM, percentage of GC and other factors. Which one of the following option represent a combination of primers that could amplify the above DNA sequence? So this is the sequence that they have given. I have just written only, I have not written all of the sequences. Only a few I have written just for the sake of primer. So here it will be, this will be one primer, 5' to 3'. This will be the other primer, 5' to 3'. So here it will be GTCATTC. So that is there in option D. GTCATTC. It is there in option D. The other primer is ATGACGA. ATGACGA. That is C. So C and D is the answer. Answer is option 4. So C is there, D is there. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of CSIR, JRF, NET, Life Science been answered, you can go to my channel Omni Bio Entrance. In the channel playlist, C for CSIR, NET, JRF, in that you will find. So that's it. Thank you.